I hope you can hear me from the other hand. Good afternoon, everyone. Yeah. Please, I want feedback. I want to know if you can hear me from your end over there.
We can hear you, please.
All right, thank you, sir. Okay. Please, can you see my screen too?
Yes, please.
I'm trying to share my screen. Can you see my screen, please?
Yes, we can.
Okay, thank you. Thank you. All right, you're welcome to the Mr. Martins by name. Yesterday I shared you some information about bioenergetics, and I told you about I explained to you about both the first law of bioenergetics and the second law of bioenergetics. So today I'll quickly take a look at some other things, but a little recap of one of the things, some of the few things I shared with you yesterday or previous days. I shared with you about that bioenergetics is just a study, a field in biochemistry that deals with the generation of energy within the cells of a living organism. So it could be an animal, it could be animal or it could be plant. So looking at it from my screen now, I shared this last 2 days or yesterday, but let me just quickly take a recap of it. I shared about the light, I shared about the types of energy that gives rise to the light energy being converted. That was the first law, the conversion of energy from one form to another. That's the law I share with you, the first law of bioenergetics. So you can see the I side, the conversion of light energy into electrical impulses. Then you can see in muscle contraction, the conversion of chemical energy to mechanical energy, and the vitamin D formation, the conversion of the light energy into a chemical energy. Then in photosynthesis, you can see the conversion of the light energy to chemical energy in plants. Like I said, that it's not limited only to human being. So the law absolutely talk about how energy can be enabled or being transferred, the possibility of energy being formed and it could be transferred. Now I want to take a look at the lesson to learn from the first law of bioenergetics. I talked to you about entropy yesterday. The entropy is a form of energy that cannot be reused in chemical reaction, but it is just the degree of energy being consumed or spread out in a system. So, and I showed you what free energy is to yesterday. So let's really take a look at it once again. Free energy is referred to as, it's the same thing as what you see in textbooks or in write-ups as gives free energy, and is usually abbreviated as capital G. So typically during energy transfer, there is a change in energy form, which is indicated by delta. That thing that you see that looks like that small capital small triangle is called delta. It's called a delta symbol. So whenever you see something like that, it means change, alteration. Don't forget, we're talking about energy transfer. There's no way you want to talk about energy transfer. Last class I explained to you why using a ice bottle water, the transfer of it when it is in solid form into a liquid. And I told you that there's a possession of energy that this solid water in a bottle has, that makes it possible for you to be in that, to maintain the solid form. So there's a requirement of energy to maintain the solid form, and there's a required energy to turn it from a solid form into a liquid form. So like I said, the free energy is referred to as Gibbs free energy and is usually abbreviated as G. Typically during energy transfer, there's a change in energy form which is indicated, that's the change indicated by that delta symbol. So thus a change in Gibbs free energy is referred to as delta G. So the change in Gibbs free energy is referred to as delta G. Quickly, let's move on. Calculating this Gibbs free energy, we have the standard free energy release. Don't forget, I told you that energy transfer means the progression of energy from a system outside, or the progression of energy from the surrounding into the system. Let me quickly use that bottle water again for an example. When you see that you have a solid bottle water, like the water within the bottle is in solid form, what happens when you start realizing that this solid form of water you have in the bottle started becoming liquid, is that the bottle water is exposed to a certain level of temperature of heat that alters the pressure that keeps it solid. So there's a relationship between pressure and the temperature, but that's not the purpose of our discussion right now. So this relationship is what causes the transfer of energy. So what happened in this case of bottled water is there's a release of certain energy that helps it to hold it together. So the more the temperature increases on the outside of the bottle, the more the energy that is required to keep it solid be released into the surrounding. I'll say it again. The more the increase of the level of the temperature on the outside or the surrounding of this bottled water that is iced, the more the increase in temperature, the more the release of the energy that is possessed by this iced water to remain solid is being released. So the latent heat of fusion is being released. The latent heat of fusion, or the latent energy required to keep it solid, is being released to the surrounding. Now I have two slides there, so let me quickly use that to express or to explain something quickly. Now the second slide where you see the temperature, you see 1 liter of water, you see the pH meter there. The pH meter is going to show the level of the acidity, the level of the level of the acidity, the level of the basicity of the compound in quotes. By looking at it now, this is a neutral, that's a pH. The pH is 7. So is that so in the right state? Now looking at it from there, you have a 25 degrees Celsius. Can you see the thermometer there? This substance is at room temperature, 25 degrees Celsius. So to calculate standard Gibbs free energy change, the formula is minus RT in products over substrate. Now, let me quickly explain something. In any chemical reaction, you have the products and you have the reactant. Is that correct? You have the product, you have the reactant. So the substrate in this case is the reactant you have. So just like, let me say you have a molecule of glucose reacting with water. So you're going to have a product on the other hand of your chemical reaction. So the product you have is the product I'm talking about here. Then the substrate you have is the reactant you have. So furthering, I have something like, what can happen to the reaction? That's the question. What can happen to the reaction? So if the products will increase, if the products increase, then we have an exergonic reaction. I'll take it again. I hope you can see my slide. If the products increase, we have it as an exergonic reaction, and you can see that the standard free energy is going to be a negative value. Then if the substrate increases, the exergonic reaction is going to be a reverse reaction. And when we have that the product and the substrate doesn't change, then we have an equilibrium reaction. If the product and the substrate doesn't change in the number of their molecules, the molecules doesn't change, there's not any change in. Now if you look at this picture you see there now, you see that the product is one molecule, is 1 mole rather. The product is 1 mole and the substrate is 1 mole. So there's no, in this case now, there's no change in the value of the molecules. So perhaps there's a change. Now you can have, you can calculate it from the formula you have there, that delta G equals to minus RT in product divided by substrate. The R is the rate of reaction and the T is the temperature. So quickly, let's take it up. Let's take it up to the next thing. Yeah. Sorry, I'm trying to adjust my slider. Okay. So you can see the change in Gibbs free energy for a reaction can be calculated with the formula you see there, minus RT in product divided by substrate. So where R is the molar gas constant, the R is the molar gas constant, then the T is the absolute temperature. The T is the absolute temperature. Then there's an information there that you should take notes. So when there's an occasion that a new equilibrium value is being attained, when there's an occasion where a new equilibrium value has been attained, it is equal to the K. That means the constant is in equilibrium. You remember the previous slide I showed you before this. Let me quickly shift it to show you more. Do you see it again? I said when the substrates and the product, and the substrate and the product does not change. You have it as equilibrium, right? So that's what we mean by this next, the next slide. I think you were given an exercise there. You can try that on your own. Time will not permit. I would have said maybe you should let me know the answer on your on the question part there. But I need to quickly move on. I've it into the time, and I want to finish on time. Now this is a table, a standard table for some of the catabolic molecules that in human. You have ADP, you have the ATP, you have the AMP, you have the glucose 6-phosphate, you have the fructose 6-phosphate, you have glucose 1-phosphate, and you can see them in the value of their free energy as shown there. Quickly, I want to go to the next slide. I want to share another thing else, and that's what I call the absolute free energy release. Absolute free energy release. Absolute free energy release. Absolute free energy release. I won't bore you with. I'll just go straight to the things that you need to know. Absolute free energy release. Now you can see there, the absolute free energy release is a modification of the Gibbs free energy. So to account for this, the substrate, the products, and the pH and the temperature, you have the formula to be the change in absolute free energy release equals to the Gibbs free energy, that Gibbs free energy, plus the molar gas constant in products divided by the substrate. Product divided by the substrate. So the change in absolute free energy release is not a constant. So as it changes continually with changes in the metabolic state of the cell. So the more there is a change in the metabolic state of a particular cell, there's going to be a change in the absolute free energy that is being released in the in the metabolic pathways. So there is always metabolism. You remember I shared yesterday that metabolism is not only it's not only about synthesis. I told you metabolism has to do with two processes. You have the catabolic process and you have the anabolic process. So yes, quickly, you can't talk about energetics without talking about adenosine triphosphate. You can see the structure there. You have a group of the ribose sugar, you have a group of the adenine, an adenine group, and 3 phosphate groups. 3 phosphate. So what makes a structure of an adenosine triphosphate is a ribose sugar, an adenine, and a 3 phosphate group. Quickly, the demand of adenosine triphosphate that stimulates exercise. So you can see the structure of the physiology of how the operation is there. You can see the reaction of adenosine triphosphate with adenosine deoxyphosphate in presence of hydrogen with it that produce sweating and dehydration. The same thing as the formation of lipids and protein catabolism. You have it in case of mitochondrial stimulation and oxygen delivery that cause the blood flow in human being, and you have the vascular functions that helps the heart functions. You have it in motor unit recruitment and neuroendocrine regulation, and you can see the reaction down there showing you the reverse reaction between adenosine triphosphate and adenosine deoxyphosphate. So quickly, let's take a look at the lesson, learning the second law of bioenergetics quickly. All reaction proceed in direction of entropy by a release of free energy. You still remember the law I told you yesterday about that. So the more negative in changing Gibbs energy, the greater the release of the free energy during a chemical reaction. Number 3, chemical reaction that has a negative change in Gibbs free energy are termed to be hexagonic reaction. Then by convention, reactions that require free energy input to proceed are termed endergonic reaction, but there are no such reaction in human body. So the free energy not used to do work is expressed as it's the free energy that is not used to do any form of work is being emitted by the body to the atmosphere as it. So to round up for today, let me share you the last information I have here. Reaction that has no net change in substrate or product are termed equilibrium reaction. I showed you. Become about that. Sorry, I showed you the other time. All reactions are potentially reversible. The directionality and the amount of free energy release of a chemical reaction can be modified by altering the substrate and the product concentration. So if you want to modify the reaction, or you want to modify the free energy that is going to be released or required for a metabolic process to happen, what you can do is you can increase or reduce the concentration of the substrate, or you alter the concentration of the product. So the increase in products may reverse the direction of the reaction. Then the increase in substrates can make the change in the Gibbs energy rather to be more negative. Don't forget, calculation in your value for Gibbs energy is going to be a negative value. Gibbs energy is going to be a negative value. So if you do your calculation and you get a positive value, then you might, maybe there's a mistake in what you put all together. So of course, if the reaction is reversed, what were the products and now the substrate and the vice versa. So on this note, I would like to take just probably a question. You have something to ask so far that I've shared with you. You can quickly drop it on the question area there. Let me know before we round up for today. Do you have any question at all? Any question? Any question? Any question? Okay. If there's no any other question, I think we could call it a day from there. So thank you so much for joining, and have a wonderful day. Thank you. Bye.